The overall goal of this procedure is to efficiently trap and image calcium transients in male C. elegans head neurons upon exposure to pheromones.
This method can help answer key questions in the neurobiology field, such as understanding sex-specific neural circuit regulation.
The main advantage of this technique is that male C. elegans can now be imaged efficiently in a microfluidic trap device.
Generally, individuals new to this method will struggle because loading animals can be difficult at first, and biogenic pheromones don't elicit calcium transients in every animal.
To begin assembly of the reservoirs, prepare 3 sets of tubing by inserting a needle into one end and a metal tube into the other. Then, for each reservoir, take a 3-way Lawler valve and attach a 30 ml syringe with its plunger removed, a 3 ml syringe, and the tubing. Fill each reservoir with the appropriate buffer, either stimulus S-basal or S-basal containing fluorescein. Then, remove any air bubbles from both the reservoir and tubing by flicking and pumping the syringe. Now, fill a new 3 ml syringe and tubing with S-basal and insert it into the outlet port of the microfluidic device. Gently apply a little pressure to the syringe until the buffer appears at the surface of the inlet holes. Next, connect the flow control, buffer, and stimulus reservoir tubes to their appropriate inlet holes, ensuring that liquid drops are present on both the loading porthole and the tubing to be attached. Now, gently apply a